7.3 Mutation. Lesson Objectives. Identify causes of mutation. Compare and contrast types of mutations. Explain how mutations may affect the organisms in which they occur. Vocabulary. Chromosomal alteration. Frameshift mutation. Genetic disorder. Germline mutation. Mutagen. Mutation. Point mutation. Somatic mutation. Introduction. A change in the sequence of bases in DNA or RNA is called a mutation. Does the word mutation make you think of science fiction and bug-eyed monsters? Think again. Everyone has mutations. In fact, most people have dozens or even hundreds of mutations in their DNA. Mutations are essential for evolution to occur. They are the ultimate source of all new genetic material in a species. Although most mutations have no effect on the organisms in which they occur, some mutations are beneficial. Even harmful mutations rarely cause drastic changes in organisms. Causes of mutation. Mutations have many possible causes. Some mutations seem to happen spontaneously without any outside influence. They occur when mistakes are made during DNA replication or transcription. Other mutations are caused by environmental factors. Anything in the environment that can cause a mutation is known as a mutagen. Examples of mutagens are pictured in figure below. For a video about mutagens, go the link below. Types of mutagens include radiation, chemicals, and infectious agents. Do you know of other examples of each type of mutagen shown here? Types of mutations. There are a variety of types of mutations. Two major categories of mutations are germline mutations and somatic mutations. Germline mutations occur in gametes. These mutations are especially significant because they can be transmitted to offspring, and every cell in the offspring will have the mutation. Somatic mutations occur in other cells of the body. These mutations may have little effect on the organism because they are confined to just one cell and its daughter cells. Somatic mutations cannot be passed on to offspring. Mutations also differ in the way that the genetic material is changed. Mutations may change the structure of a chromosome or just change a single nucleotide. Chromosomal alterations. Chromosomal alterations are mutations that change chromosome structure or quantity. They occur when a section of a chromosome breaks off and rejoins incorrectly or does not rejoin at all. Possible ways these mutations can occur are illustrated in figure below. Go to this link for a video about chromosomal alterations. http://www.youtube.com/watch?v=ORXRSQA_3LU, 218. Chromosomal alterations. Chromosomal alterations are major changes in the genetic material. Chromosomal alterations are very serious. They often result in the death of the organism in which they occur. If the organism survives, it may be affected in multiple ways. An example of a human chromosomal alteration is the genetic disorder known as Down syndrome. It occurs when an individual possesses a partial or complete third copy of chromosome 21, leading to developmental delays and physical abnormalities. Point mutations. A point mutation is a change in a single nucleotide in DNA. This type of mutation is usually less serious than a chromosomal alteration. An example of a point mutation is a mutation that changes the codon UUU to the codon UCU. Point mutations can be silent, missense, or nonsense mutations, as shown in table below. The effects of point mutations depend on how they change the genetic code. Type description. Example effect. Silent mutated codon codes for the same amino acid CAA, glutamine, CAG, glutamine, none. Missense mutated codon codes for a different amino acid CAA, glutamine, CCA, proline, variable. Nonsense mutated codon is a premature stop codon CAA, glutamine, UAA, stop, usually serious. Frameshift mutations. A frameshift mutation is a deletion or insertion of one or more nucleotides that changes the reading frame of the base sequence. Deletions remove nucleotides, and insertions add nucleotides. Consider the following sequence of bases in RNA. OG AAU ACT GCU equals start asparagine 3-anine alanine. Now, assume an insertion occurs in this sequence. Let's say an A nucleotide is inserted after the start codon OG. OG AAUACG GCU equals start lysine tyrosine glycine. Even though the rest of the sequence is unchanged, this insertion changes the reading frame and thus all of the codons that follow it. As this example shows, a frame shift mutation can dramatically change how the codons in mRNA are read. This can have a drastic effect on the protein product. Effects of mutations. The majority of mutations have neither negative nor positive effects on the organism in which they occur. These mutations are called neutral mutations. Examples include silent point mutations. They are neutral because they do not change the amino acids in the proteins they encode. Many other mutations have no effect on the organism because they are repaired before protein synthesis occurs. Cells have multiple repair mechanisms to fix mutations in DNA. One way DNA can be repaired is illustrated in figure below. If a cell's DNA is permanently damaged and cannot be repaired, the cell is likely to be prevented from dividing. DNA repair pathway. This flow chart shows one way that damaged DNA is repaired in E. coli bacteria. Beneficial mutations. Some mutations have a positive effect on the organism in which they occur. They are called beneficial mutations. They lead to new versions of proteins that help organisms adapt to changes in their environment. Beneficial mutations are essential for evolution to occur. They increase an organism's chances of surviving or reproducing, so they are likely to become more common over time. There are several well-known examples of beneficial mutations. Here are just two. Mutations in many bacteria that allow them to survive in the presence of antibiotic drugs. The mutations lead to antibiotic-resistant strains of bacteria. A unique mutation is found in people in a small town in Italy. The mutation protects them from developing atherosclerosis, which is the dangerous buildup of fatty materials in blood vessels. The individual in which the mutation first appeared has even been identified. Harmful mutations. Imagine making a random change in a complicated machine such as a car engine. The chance that the random change would improve the functioning of the car is very small. The change is far more likely to result in a car that does not run well or perhaps does not run at all. By the same token, any random change in a gene's DNA is likely to result in a protein that does not function normally or may not function at all. Such mutations are likely to be harmful. Harmful mutations may cause genetic disorders or cancer. A genetic disorder is a disease caused by a mutation in one or a few genes. A human example is cystic fibrosis. A mutation in a single gene causes the body to produce thick, sticky mucus that clogs the lungs and blocks ducts in digestive organs. You can watch a video about cystic fibrosis and other genetic disorders at this link, http://www.youtube.com/watch?v=8s4he3wlgkm 931. Cancer is a disease in which cells grow out of control and form abnormal masses of cells. It is generally caused by mutations in genes that regulate the cell cycle. Because of the mutations, cells with damaged DNA are allowed to divide without limits. Cancer genes can be inherited. You can learn more about hereditary cancer by watching the video at the following link, http://www.youtube.com/watch?v=LWK5FPLSKWM 429. Albino redwoods, ghosts of the forest. What happens if a plant does not have chlorophyll? They would lack the part of the leaf that makes them green. So these plants could be referred to as albino. This would have to result from a genetic mutation. Do these plants die because they cannot photosynthesize? Not necessarily. What can these plants tell us about the biochemistry, genetics, and physiology of plants?